Namaste. I'm Dr. Jayoti from Department of Food and Biotechnology, Agriculture and Veteran Science, Jyoti Vidyapeeth, Performance Institute, Jaipur. Today I'm going to discuss about the topic microbial growth and their aspects in measurement. How we can measure the growth of microorganisms. Uh, First, what we mean by the growth of the microorganism. The growth of microorganisms is meant by the increase in the number of the microorganisms. Means the microorganisms increase in their number from 1 to 2 to 4, 4 to 8, 8 to 16, 16 to 30. So they multiply. So this is known as the microbial growth. And similarly, the size in the microorganism uh, increase in their shape, increase in their size. So what are the techniques which are used for the measurement of the microbial cell numbers? So there are different lab manual techniques which are available for the calculation of the microbial growth. Uh, there are two types of techniques. First, direct cell counts or viable cell counts. Okay, we will discuss one by one these techniques. First is the direct cell count. The direct cell count include the counting chambers, electronic counters, on-membrane filters, and the viral plate counts include plating methods like spread plating method and membrane filtration methods. So these are the techniques. First is counting chambers. Counting chambers are very important and easily available technique in the microbial labs. It is easy. So it is easy. It is inexpensive and method. Counting chambers are easy. They are inexpensive and quick method, and they are useful for counting both eukaryotic and prokaryotic cells. So bacterial cells, fungal cells, or any kind of cells can be counted using the counting chambers. And these counting chambers cannot distinguish between the living cell and the dead cells. They are used only for the counting. They are not used for the counting of non-viable and viable cells. Next is the electronic counters. Electronic counters are known as the counter. Okay. They are known as the counter and micro suspension are forced through a small hole or a surface by which the micro can be passed. And movement of microorganism through the hole impacts the electric currents. Okay, when the microbes will pass through this hole, they will impact it electric current, and the number of the, number of microbial cell will be proportional to the and the amount of the current will proportional to the number of the microbial cells. Okay, so this technique also cannot distinguish between the cells of living and non-living cells, means viable cells or non-viable cells. The advantage is that this technique is quick and easy to use. It is useful for the large microorganisms. It is used for the distinguishing of large microorganisms and blood cells, and it is not for the prokaryotic organism, it is for the eukaryotes. So electronic counters is also very important. Next is the membrane filters. Uh, what are the membrane filters? Membrane filters have a particular kind size. Suppose that there is a membrane filter of 10 micrometer. So only the microorganism of the 10 micrometer microorganisms smaller than the 10 micrometer will pass through the membrane filters. But the microorganisms which are larger than 10 micrometer will not be allowed to pass through the filters. Okay. There will be separate membrane filter for a separate membrane filter and different size lines for different kind of membrane filters. So in the membrane filter, cells are filtered through special membrane that provide dark background or absorbing. Okay. And cells are stained with the fluorescent dye, and this is useful for the counting of bacteria. And there are different type of dyes can be used, which can distinguish between the living from dead cells. Okay. So these are the techniques which are used for the measurement of the microbial number. Now discuss about the important environmental factors which affect microbial growth. What are the different environmental factors? First one is the pH, temperature, gas, pressure, and other environment. So we will discuss each one. Firstly, the pH. pH is the negative logarithm of hydrogen ion concentration, and pH affect the activity and integrity of enzymes and structural component of a cell. So optimum temperature which is required for most of the microorganisms is approximately 6 to 8. Okay. Acidifies fluid 0 to 5.5 pH, and neutralifies flow between 5.5 to 7 pH, and alkalophiles grow between the 8.5 and pH 11.5, between pH 8.5 and 11.5. So these are the different pH requirement for different kind of microorganisms. Now the effect of temperature. Organisms exhibit distinct cardinal growth of temperatures like minimal temperature, maximum temperature, or optimum temperature. So some microorganisms are psychophiles, some are mesophiles, and some are thermophiles. Uh, Psychophiles grow at can grow at 0 degree centigrade, grow optimum at 15 degree centigrade or lower, and they can survive cold environment. Okay. But they cannot grow above 20 degree centigrade. Psychotrophs are the organisms which can grow at 0 to 7 degree centigrade, and they can grow max to max 35 degree centigrade. And the mesophiles are the organisms which grow optimum around 20 to 40 degree centigrade. Moderate temperature of these organisms will differ. Now thermophiles are the organisms which grow at higher than 45 degrees or more temperature, more than 45 degrees centigrade, and they can also tolerate a temperature of approximately 80 degrees centigrade. So they grow between the 45 and 80 degrees centigrade, and the optimum temperature of these thermophiles 55 to 65 degrees centigrade. Yeah, he can microorganisms. The thermophiles are heat living microorganisms. Next is the hyperthermal bias. The optimum goes between 80 and 100 degrees centigrade and the temperature of about 120 degrees centigrade. Now, gas requirement of the microorganism. Depending on the kind of the microorganism, there are different kinds of oxygen requirement in per organism. Uh, like aerobic microorganisms like Pseudomonas aluminans need high oxygen concentration. Facultative aerobic which badly need oxygen for their growth, and E. coli and obligate anaerobic like Clostridium butyricum do not need die in the presence of the oxygen. Obligate aerobe need oxygen. Facultative anaerobe prefer oxygen for their growth, and an aerotolerant anaerobic can ignore oxygen, and strict anaerobe they do not need oxygen, and oxygen is toxic to these strict anaerobic, and microaerophilic require 2 to 10% of the oxygen concentration. They do not need high concentration of oxygen. Now, pressure, effect of pressure. Microbes obtain almost all nutrients in the solution from environment, and verified organisms are the organisms which are adapted to the high pressure conditions, and they require or grow more rapidly in the presence of increased pressure. Okay, means the microorganisms live in the deep sea are aerophiles, means they grow at high pressure conditions. So these all are the about the microbial growth, in which I discuss about the what are different requirement of the microorganism, what are the different techniques for the counting of the microbial growth. Okay, and I also discuss what are different factors which affect, what are different environmental factors which affect microbial growth, including the pH, temperature, gas, pressure, and some environment factors, radiation light. So I hope the students will better understood all these things. So what kind of questions which can be asked? The examiner can also ask you that explain the different techniques for the measurement of the microbial growth. Okay, so you can answer it, and they can also ask you what are the different factors in the growth of the microbial growth. Okay, so you will better answer all these questions. So my lecture is over here. If you have any query, you can ask me. So thank you very much.